Hi there. So today I'm going to explain you how to run our beta amylase assay for Malt. Here we have a 6-pack that we have sent you, which contains 6 beta amylase kits, and you can see here the QR code that you will use specifically for this kit, and you can also read that there's a beta amylase kit in this QR code part. We also have here a series spectrophotometer. As you can see, it has the lights on. So this is what we have in each kit. We have a vacuum tube with 25 milliliter buffer. We also have a series of chemicals, mostly a green sticker for the extraction powder, red sticker for the stopping reagent, a brown tube for the substrate. We also have all the plasticware that you need. Basically, you have a 5 milliliter empty tube. We'll see later what we use it for. A syringe. We also have cuvettes and a couple of Pasteur pipettes. The first thing we need to do is to actually pre-weight your mole sample. Add the extraction powder in the buffer. At the Glycospot, we pre-weighted the powder for you, so you don't need to worry about that. You add it. Make sure that if there's a last part, you put it. And then you shake it for around 5 seconds until everything is dissolved. It dissolves very quickly. And then the next step, add your mold into the buffer. Make sure that everything is inside. And then you shake it for just 5 seconds to make sure that it's homogeneously distributed in the extraction. Timer pipped, and what we will do is we take the tube out. We can also take the advantage to take the whole holder out of the heating box, and then we place the new holder, or the holder for the epender tubes. What we have here is an extraction of the malt, so the enzymes are now in the liquid, but what we need to do is to separate the most liquid part from the particulate that it could have. So we use the syringe that is included in the kit. We take around 5 milliliter, it doesn't need to be very precise, and then what we do is we filtrate around 5 milliliters of the solution. Now we have a clear solution with the enzymes of interest, in this case beta amylase, and what we will do is to incubate these enzymes with our own substrate. So we can place the tube here in a support that we give in the kit, and what we need to do is to pipette 250 microliters from this tube to the substrate tube. You just need a regular 250 microliters pipette, or a pipette that can measure that volume, and then you would just go ahead and pipette this volume from the filtrate to the substrate, and quickly, or as quick as you can, take it, mix it, and place it in the heated incubator. Now it needs to incubate for exactly 7 minutes, or as close to 7 minutes as you can, because we are measuring an enzymatic kinetics, so time is critical in this measurement. So in the meantime, where this is incubating, what we're going to do is we're going to prepare what we call the reference of the sample. That is to cancel any color of the sample. So what we will do, we'll take the empty small transparent tube, and we'll place 250 microliters of the solution and 250 microliters of stopping reagent, which is signed in or with the red circle. So we're going to take 250 microliters of this and place it with the other liquid. You do is you mix it for around 5 seconds so as to make sure that it's mixed. All what you need to do is to transfer this mixture into one of the 2 cuvettes that you receive with the package. So after the 7 minutes, you take the tube out from the heated incubator, you place it here, and as quick as you can, you add the stopping reagent. Again, 250 microliters of stopping reagent. Close it. What you need to do is a few mixing of, again, around 5 seconds, and that is ready to be read in the spectrophotometer. So now we have the tube with the stopping reagent, and what we need to do is to transfer this into a cuvette. So we open it, and with the help of a Pasteur pipette, first from the lid, then from the bottom, we take the resulting liquid. And now we are ready to read this with our spectrophotometer series. In order to measure with our series spectrophotometer, what you need is to open the app, BrewerStream. As I mentioned, you can download it from App Store or Google Play Store. And the first page that it appears is to scan the devices. This device needs to be turned on to be able to read it. In this page of the app is to scan the QR code by pressing to this button, and then scanning the QR code. So the first step in the app is to actually add the reference samples that we prepared as a mix between stopping reagent and filtrate. You will press measure, and then we can give the sample an ID, which could be, for instance, number 1. We save, and then the next measurement is actually the color resulting from incubating the sample. We press measure again, and this is the result, 254 Vindish Cobalt, which is very close to the reference value, which is around 250 Vindish Cobalt. So that was pretty much it when it comes to the assay. It's very simple and easy to use, but if you have any doubt or any question, please don't hesitate to make us a call or send us an email. We are very pleased to help you, and we will be there to answer any of your questions. Thank you.